Welcome, my name is Justyna and I would like to present the most popular methods used for isolation and cultivation of microorganisms. Why microalgal strains are isolated from environmental samples and cultivated in laboratory conditions? Clonal cultures of microalgae are used for basic research and in different sectors of biotechnology. In this presentation, I will demonstrate one of the isolation techniques. In some cases, it is easy to isolate and cultivate a strain. However, some species are difficult or thus impossible to isolate and culture. Knowledge about specific requirements of the organisms is the first step to successful isolation of microorganisms. To properly isolate microalgae and start cultivating them, you need basic laboratory equipment such as Pasteur micropipette, petri dish, microscope slide, multi-well plate, test tube, culture flask, burner, different media also with agar, small loops, forceps, pipette, tips and ethanol. It is also important to choose the right microscope. The dissecting microscope has enough space to work with a micropipette, but on the other hand, it doesn't allow you to magnify the object properly, often making it impossible to see a single cell. Individual cells are well seen with a light microscope. However, here, the working distance between the lens and the samples is too small, making it difficult to pick cells with a micropipette. Therefore, the inverted microscope is most commonly used for microalgae isolation. There are a few standard methods used for microalgae isolation. The most popular are dilution technique, isolation on plate with agar, and single-cell isolation by micropipette. Enrichment cultures are used as a preliminary step towards single-cell isolation. They are established by adding nutrients to the collected phytoplankton samples. The dilution technique is only applicable in the case of abundantly occurring organisms. The goal of this method is to have only one cell or filament in the test tube, flask or well. Usually, you inoculate a large number of tubes or wells with the most diluted sample, assuming that some single cells may die, some tubes or wells will contain two or more species, whilst some will receive no cells at all. Remember that the dilution technique doesn't allow you to obtain arsenic isolates. To remove bacteria, a proper antibiotics should be added. Alternatively, you can isolate your culture on agar plates. This method is mainly applicable for coccoid algae and most of the soil algae. Isolation is accomplished by streaking the water sample across agar surface. With the use of a bacterial loop, you should collect a small volume of water sample and spread it across the agar surface. The first streak usually has too many cells that are not well separated. But as the distance from the first streak increases, single cells become more and more visible. After streaking, incubate the agar plate until distinct colonies appeared on agar surface. The incubation time varies depending on the species. When colonies originating from a single cells appear, you should transfer them to the new agar plate or liquid culture medium. Remember, not all species can grow on agar. Isolation with a micropipette is the third method I would like to present, and at the same time it is the most commonly used one. The method is usually performed with a Pasteur pipette or glass capillary. The goal of micropipette isolation is to pick a single cell from the sample, deposit it into a sterile droplet of medium, pick the cell again, and transfer it to a second sterile droplet. This cleaning process should be repeated until in a droplet a single alga cell will be found. Finally, the last drop with one cell is transferred to well with a culture medium. In case of filamentous cyanobacteria, the same procedure is followed for a single filament. Incubate the plate for several days under conditions optimal for the growth of the isolated strains. Control the growth of the isolated organisms. Transfer the selected strain to a vessel of higher volume and then incubate the strain under controlled conditions. Culture conditions should be optimized for each isolated strain individually. Under favorable conditions, the biomass doubling time for microalgae during exponential growth can be as short as 3.5 hours. Thank you for your attention.